Hello and welcome back. So now that we're familiar with the central dogma of biology, we understand the significance and key regions of our DNA and RNA, and we know the various ways of controlling gene expression without altering the sequence of DNA, now we're going to enter the exciting DNA-altering world of mutations. Mutations are defined simply as a change. With DNA, that means a change in the nucleotide sequence. We call the four letters of DNA, G, A, T, or C, nucleotides. Simply changing or having mutation of one or more of these DNA nucleotides can lead to a change in the mRNA and ultimately a change in the protein that is made. Some possible outcomes from a DNA mutation are a non-functional protein, a protein that has partial function, a protein with a completely new or novel function, no protein at all, or interestingly enough, no noticeable change in protein function even though there was a DNA mutation. Mutations of DNA may also affect timing or level of gene expression. One of the fundamental properties of mutations is that mutations just occur randomly. When and if mutations occur is completely unrelated to any advantage that mutation may provide. They don't arise because an organism needs them, and conversely, they don't arise to harm an organism. They're spontaneous. Natural selection plays a role in these mutations, whether they'll exist in a population or not, because they can be selected for or against. Now, I want you to think about DNA and RNA as sentences that code for genes. All the genes in the sentence are composed of three-letter words that we're going to call codons. In the same way, three letters can make up a unique word. Three nucleotides make up unique codons. The nucleotides for making up DNA codons, again, are A, G, C, and T for DNA. For RNA uses the nucleotides U instead of T. So the four nucleotides of RNA are A, G, C, and U. Three-letter codons correspond to specific amino acids, and those amino acids, to refresh your memory, are the building blocks of proteins. In this way, the three-letter codons determine which amino acids are going to be arranged in the proper order to synthesize the proper protein. So we can think of codons that make up a gene as a sentence comprised of three-letter words. One example of a sentence comprised of three-letter words is, the red hat was big. You'll notice that this analogy corresponds to a gene in that we have a sentence composed of three-letter words. Again, the red hat was big. Two types of mutations we're going to talk about are insertion mutations and deletion mutations. And you can probably hypothesize from their names what happens in each mutation. For an insertion mutation, one or more nucleotides will be inserted. A deletion mutation will have a deletion of one or more nucleotides. These mutations, insertions and deletion mutations, are called indels, and they can lead to something called frameshift mutations. Consider our sentence, the red hat was big. In the case of an insertion mutation, imagine we insert a letter, say A, into the word red. So now when you try to read the three-letter words, it reads the Rhea. Well, you'll notice that's not a word, and everything else in the sentence is gibberish. What effect do you think that would have if we changed the codons used as a template to make a protein in the same manner? Now imagine the other type of mutation, the deletion mutation. You have the same sentence. The red hat was big, but in this case, the D is removed. It's deleted. We have the wait, that doesn't make sense. Those three-letter words don't mean much. So we have a frameshift mutation occurring again, since RNA is read in three-letter frames. Once the word is altered, one word is altered, the rest of the sentence is also shifted. Another type of mutation that we'll talk about are called substitution mutations. These don't change the reading frame of the sentence. We have the same sentence, the red hat was big, but for a substitution mutation, we'll substitute, in this example, an E in red with an A. So now we get a different sentence with a different meaning. The rad hat was big. I actually introduced this type of mutation earlier when we discussed the sickle cell trait. In that case, one single codon is changed by substituting one nucleotide with another nucleotide. And you can see the dramatic effect it had on the shape of hemoglobin. It altered not only the shape, but remember, form dictates function. A change in the shape of hemoglobin changed how it functioned. If we want to look at gene mutations, it's difficult to study in humans, but because we never have control of a human's environment or lifestyle. And when we try to study DNA mutations in humans, we often run across that pesky issue of ethics. So fortunately, we have model organisms that we can use to study mutations. We'll see that nature's thrifty and that a lot of genes do the same thing in flies, mice, and even tiny bacteria. One example of this similarity in different organisms can be seen in people with a mutation called the Tin Man gene. It's named after the Tin Man in the Wizard of Oz, and people with this mutation often have congenital heart defects. But this mutation was first observed in fruit flies born without a functional heart, hence the name Tin Man gene. Based on the similarities in gene function and expression in flies and humans, you can see why some geneticists refer to flies as little humans with wings. Flies drink alcohol when they're sexually rejected. They can become aggressive, so we can use them to study behavior. They're great models because they have sleepless nights and they can be used to study insomnia. They're a wonderful model organism because we can study these traits and much, much more. I want to drive home the connection between form and function so that you truly understand significantly how a little change in our proteins can ultimately affect our overall body's function. So we talked about form and function. I want to give you a practical example of form and function. Take this chair, for example, a very specific form or shape that relates to its very specific function. What if I change the shape or form ever so slightly? So, just change where the seat is. Has it affected the function? Well, let's see. I can sit on it. It's asking for a mishap, but I can still sit on it. So a slightly different shape, slightly different function. It's not going to work in the same way. But let's look at it more dramatically. What if there were a dramatic change in the shape of this chair? So now I change the shape. All the elements are still here, but is the function the same? No, the function's not the same. So what we're going to see when we talk about proteins and mutations is that different changes in shape can have different effects on the function. Some can be small or non-consequential, and other ones can be pretty extreme. Now imagine if I were to start hitting this with a hammer. That's going to have a much more extreme effect on the shape and a much more extreme effect on its function. Although mutations are often thought of as harmful, they provide a rich source of genetic diversity in the world around us. Mutations can play a key role in why you don't look exactly like your mother or your father, sister, or your brother, but why you're uniquely you.